So a virus is an organism without, uh, like, they don't have many organelles which are present in a normal cell. So that's why virus, like I believe most of you know that it can be like, normally we say virus either in non-living phase or living phase, right? So those two phases exist. That means when virus, they are present outside the cell, they are non-living particle. But when they are going inside the cell, then only it shows its activity. That means it can make more viruses, and finally it can increase their number. But a virus outside the host cell, you can say that it's a non-living particle. So now the question is, this is the only case where we are saying that this is in between living and non-living thing. So I will discuss why it cannot stay as living thing outside the cell. If I ask you that, do you know what is the major reason that why virus cannot stay as a living being outside the host cell? What will be your answer?
So they don't have any protein forming mechanism on their own. They have to depend on the others. Like the
Yes.
machinery is not present in their cells. You said the organelles are not present, so they have to
Yeah.
depend on the others host cell for that. So as soon
Very
as they come out the host cell, the cannot produce the proteins and do that.
Mm. Very good, Tanmay. So that means virus, they have their genomic material, they have genes, they have some of the important proteins also. But the problem is they don't have protein producing machinery. The major thing missing in viruses is ribosome. They don't have ribosome. So they cannot translate their own thing. So as a result of that, when virus enter inside the host cell, then they actually utilized host machinery, host ribosome, and many other things. Many other things are missing, but ribosome is one of the important thing which can translate protein, right? But that is also not there. So as a result of that, only when virus is going inside the cell, then it can utilize host ribosome and many other accessory thing in order to make more and more protein as well as more and more genomic material so that more virus particles can be generated. So that's the reason why it should be inside the cell. So whatever mentioned here in this diagram in the living phase, so I will discuss this part later, like in some other slide. So that's why we put viruses into a separate kingdom of life. Kingdom means, see, normally we say we have plant, animal, and archaea, bacteria, this kind of kingdom, but viruses could be considered as the simplest, hence ancient form of life. But I put here this, sign because whether it's life or not life, that depends on whether viruses they are staying outside or inside the cell. Composed of nucleic acid and proteins, as I already told, and a virus is an infectious obligate intracellular parasite. Just clarifying this term, infectious, it will infect host cell. Obligate, that means it must go inside the host cell. Otherwise, it cannot either grow or it cannot survive. So obligate intracellular parasite, that means it's go inside the cell. It should go inside the cell. Like, so for example, like why inside the cell? I'm just telling you, say, for example, in human, we are multicellular organism, we have many cells. So in between cells also we have space. So some bacteria, they actually infect in that way. So many bacteria, they are not intracellular. So this is apart from virus, but I'm just trying to explain you the overall idea. Some bacteria, they can infect us, but they are not going inside the cell. They are staying outside the cell, in between cell. We have lot of space in our body in between cells. Those are called cell matrix, cell junction. That is not inside the cell because by bacteria they can survive by their own. They are only taking food, nutrients. That's all because they have all other organelle required for survival. But viruses, as I asked you and Tanmay just few minutes back replied, that virus need to use host machinery. That's why viruses are obligate intracellular. So it must go inside the cell. So that's the reason. Now, here, virus can exploit all three domains of life, as I told you, archaea, bacteria, and eukaryotes. So here, archaea, this term you might not heard before. This is little bit primitive to bacteria. And bacteria, all of you know, like E. coli and many other bacteria we have. And eukaryotes means here plants and animals. Okay, so whatever organism present in our earth, we classify in these three different kingdom: archaea, bacteria, and eukaryotes. But viruses, you cannot put virus in any of these three, so that's why it is in between living and non-living particle. But let's see how it infect and what it does. So what is a virus? First thing is virus is a submicroscopic particle. Many things are submicroscopic. Even a bacteria is submicroscopic. But the thing is, the viruses are much smaller than bacteria, and only you can see viruses through electron microscope. It is not possible to see viruses through light microscope. Okay, and here we are just showing some of the viruses like bacteriophage. I mentioned before actually in genetic engineering classes that bacteriophage infect bacteria, right? So it looks like this. Most likely you studied in your school days. Coronavirus, this is very common one, right? It is there for a long time. Whatever coronavirus you are listening now, that is some kind of new variant, but coronavirus is very common and it is there for from long time. And influenza virus, and here you can see Ebola virus. This is a very dangerous virus, but it looks like completely different kind of thing, kind of filamentous cell. So this is called Ebola virus, and all these images are real images, transmission electron micrograph of virus particle. So here in the
So
lower panel,
this
yeah.
coronavirus, this coronavirus picture is of the new variant or of the old
No,
variant?
no, old variant. It is like I believe around 1965. During that time, even people actually tried to capture images of coronavirus, like coronavirus through electron micrograph. Okay, because in this resolution, whatever I'm showing that it's transmission electron micrograph, you didn't see any difference between like new coronavirus, like whatever the variant now, or the older one. Okay, so you will not see any differences. It will be similar. But in order to see even more difference, then you have to actually, get the structure. Say, for example, in current coronavirus, all of you are aware about spike protein. So then you have to crystallize this spike protein. You have to get the structure of spike protein. So I have some of those slide at the end actually. Okay, so you will get like at the at the end I will concentrate on SARS-CoV-2 also. So here in the lower panel, you can see a virus comprises of genetic material which is surrounded by a protein coat. So here, this is genetic material in red color, and this in a cyan color beads, those are the protein coat. And in viruses, what happens? Like sometimes a single type of protein can be covered, or multiple proteins can be there. I mentioned during protein class that rhinovirus in their coat they have multiple protein and they are organized in a very, I would say, in a higher order, and I told you that that is a good example of protein quaternary structure. But some viruses they have just one type of protein and that make the protein coat. And here you can see that same type of protein, but multiple protein subunits should be there. And this protein coat is called capsid. This protein coat is called here capsid. And each unit, that means every protein present there, those are called capsomere. So this is some kind of terminology. If you forget it, nothing will happen. But don't forget this capsid term. This is required, right? This protein coat is called capsid. And some viruses, they even have another layer that is called envelope. So this green color stuff you can see, this is envelope. Okay. So now this envelope is
So the
actually,
capsomer is each unit of the capsid, a capsid.
yeah, exactly. That means those are protein actually, like separate protein. And all of them together, they form the capsid, that protein layer, which is present outside the virus, like the protein coat. Okay, yeah. And some viruses, even outside the capsid, they have another layer that is called envelope. This is actually, derived from host cell itself. This is a little bit complicated. I'm trying to explain you. So virus, they don't have information to make their own envelope. Envelope means some kind of lipid layer, but that is present outside the capsid layer. Then how they acquire this envelope? Because when viruses are growing inside the cell and they are coming out from the cell, during that time they take some of the membrane portion from host cell and that surround those virus particles. And then what is the utility? So do you know what can be whether it will be helpful for viruses or not? It is actually sometimes helpful because like
Resistance.
uh, hmm, someone want to say.
No, sir, I was saying that it can provide thermal resistance or resistance to certain chemicals.
Yeah, so definitely those that is more resistant compared to protein. It's true. In addition to
So,
that if yeah.
also, like there will be like if the immunity cannot recognize the virus cells anymore because it is now surrounded by the
Oh, yeah, that's
membrane.
yeah, I understand. That's a good perspective actually, but I'm not sure, Tanmay, that whether it's really work or not because whenever it will go inside the cell, it will disassemble, and that time our immune system might like it should recognize. So, but it's a good perspective actually. Like I would say, good thought. So I will see. Like, but apart from that, also another thing is lipid and lipid they can mix together. So if they have lipid envelope, that will help to attach with the cell membrane again. That means when virus infects someone, like in some portion of your body, some virus infection happen, right? Now, when virus particles are mature inside the cell, that means from few viruses, you have a lot of viruses, they are coming out from the cell and they will again quickly infect other cells, right? Sometimes this infection might happen through some spike protein, sometimes just fusion of this membrane to host cell membrane. So as a result of that, again the virus will go inside the cell and that will help dissemination of viruses in other host cell and tissues. So that is another advantage of viruses having envelope. But many viruses they don't have envelope. Most of the viruses they have capsid or protein layer. For example, in case of SARS-CoV-2, they don't have envelope. They have capsid. They have spike protein. Okay. So let's see. Viruses is a submicroscopic particle, as I already mentioned. Here you can see this one. This is a eukaryote. This is an eukaryotic cell, right? Eukaryotic cell. You have nucleus here, mitochondria, so many different organelles. And if we zoom this portion only, right? So this portion is this portion, this curvature you can see here. This is the same thing here. So you can see inside the cell there are many things there. For example, ribosome, this is ribosome. Okay, all this white thing here, ribosome, and those are microtubule, this chain like structure, and so many organelle proteins, and that is called macromolecular crowding. I told before also that inside the cell it's not like everything in solution. It's kind of lot of protein, many things are there, and they are always in Brownian motion, but still every biological functions are happening in right way. So now if you see the dimension of viruses is even very close to ribosome. Thus, like for example, poliovirus. Poliovirus is actually very small in size, and their diameter is 30 nanometer. compared to, like, if you see herpes virus, they are a little bit bigger. So their diameter is 20 nanometer, right? And more like, most of the other viruses in between these two, like from 30 nanometer to 200 nanometer in diameter. So that means viruses are very, very small actually. That means many viruses can be present inside one cell. Like huge number of viruses can be present inside one cell. As I told you before, that in a healthy cell, inside the cell, you might have more than say 25,000 ribosome, 30,000 ribosome. So you can estimate that virus can be also in huge quantity inside just one cell, right? So one thing, just I'm stopping. So what about the like sound quality, the internet connection? This is same like previous classes, or little problematic?
So it is okay.
Okay, very good. Unique features of virus. Here, some interesting thing will come. So, virus possesses DNA or RNA as their genetic material. Uh, you might know from your, like, even from newspaper, from literature, from news channel, that this SARS-CoV-2, they contain RNA as their genomic material, right? So virus, some other viruses, they also have DNA as their genetic material. And virus, they lack ribosomes or any membrane-bound organelle. Virus hijacks host cell machinery for their own good. That means to make more and more virus particle. But here, I would like to summarize some previous concept. Like I told you all the time that DNA is our genetic material and RNA is, operational. That means the RNA just connect between DNA and protein, right? Then virus, they are only organism if you consider that they are organism when they are present inside the host cell. They are only organism in the earth to contain RNA as their genetic material, right? Then the biggest question is how the flow of genetic information in viruses fits, which with the concept of central dogma, whatever I discuss with you several times, that DNA goes to DNA, then you can get RNA from DNA, that is called transcription, and then you will get protein from RNA. This is the flow of genetic information. Then if they have RNA as their genetic material, then how you can address this issue? So who will answer this question? At least try to answer this question.
Sir, an enzyme called reverse transcriptase
Hmm.
performs reverse transcription and converts RNA to DNA, and then the
Okay.
normal central dogma of life happens.
So that is that also that can be fitted in central dogma, right?
Yes, sir.
Yeah.
So, but the reverse transcriptase that we use, that is artificial, right? It's not uh, natural.
So yeah, that means viruses, they have their own information to make reverse transcriptase. Okay, so virus have genetic material. In their genetic material, when that virus will come inside the cell, that will make, like, from that genetic material, host cell will make some protein, and that one of those protein will be reverse transcriptase, so that they can synthesize their, they can make DNA out of the RNA. Right? I will discuss that. Let's see. So
Okay, sir.
now, yeah, if you see, that's why in every textbook, mostly you will see that this is flow of genetic information, and even sometime people say that genetic information, the flow of genetic information is not correct. The reverse transcription is there. What Rajas just told, that from RNA to DNA can be generated. Even RNA to RNA can be possible even in viruses. So it needs that, we need to revise the central dogma, but that is not true. Actually, in central dogma, Francis Crick proposed all these possibility. I will tell you. So here, this is flow of genetic information, but some additions are there. But this was not addition actually, I would say. People discovered that yes, those things are true, but long time back, Crick actually proposed the same thing. So one thing is reverse transcriptase. One particular enzyme, RNA polymerase that makes RNA out of DNA. A reverse transcriptase just opposite. It uses RNA as template and it makes DNA, and that is called reverse transcription. You can see just it is going in reverse direction. So this is reverse transcription. Okay, and you might know that David Baltimore, he actually discovered reverse transcriptase, and most of his life David Baltimore worked on HIV viruses, and he got Nobel Prize also for the discovery of reverse transcriptase. Okay, and at the similar time, some other scientists, they also discovered that it is also possible to make RNA out of RNA. So RNA out of RNA, just like DNA to DNA, you can make RNA out of RNA. So the enzyme involved here is called RNA-dependent RNA polymerase. So here I would say try to remember this name, because those names are not nothing something like that you cannot remember, because in this case the transcription is opposite of transcription. That's why reverse transcriptase and AAC present after enzyme name, right? Similarly here, RNA-dependent RNA polymerase. That means you know that RNA polymerase that makes RNA out of DNA. But in that case, DNA is the template. But in this enzyme, RNA-dependent RNA polymerase means it relies on RNA. RNA is being used as template. Again, this is observed in some viruses only, and reverse transcription is also observed in some viruses, right? So all those things are actually together, they make the central dogma, and you will be surprised to see that as I told you, that even some textbook mention that flow of genetic information needs to revise, and after the discovery of reverse transcriptase as well as RNA dependent RNA polymerase, it came to know that it is going in different direction. But if you see who proposed this idea of like central dogma, Crick, Francis Crick here. So this is his idea. He mentioned in 1956, right? 1956, long time back. So if you see here that he mentioned that central dogma means once information has got into a protein, it cannot get out again. So that was central dogma. So he actually mentioned the possibility of RNA dependent RNA synthesis, as well as RNA can go back to DNA also. Dotted line, he was not like sure about that, but he mentioned that it could be done, right? It is possible. So that way, viruses are fine actually, although they have sometime RNA as genetic material, so that doesn't matter actually. Sometime from RNA, they can make DNA, and again, they can make more and more virus particle. Sometimes some viruses, they have RNA, and from that RNA, they can make more and more RNA. That time they use RNA-dependent RNA polymerase. Then you can ask me that why these two possibilities are there, because some complexity there. I'm not going into details, but I would like to say here one thing. If you get it, that's good. Otherwise, you can actually, ignore at this level. So DNA, when I discuss about DNA in details, like I mentioned that two types of two strands there. One is called sense strand, one is antisense strand or template strand, right? Sense strand means the same information is present in mRNA, that is coding strand, right? And then mRNA, and then you are getting protein. Then if some viruses, they contain RNA, then the question is whether that RNA will be directly utilized to synthesize protein, or you have to make the other strand. That means if the virus, they contain RNA as genetic material, whether that is actual carrying the sense, or you have to convert it into a template. That means the exactly complementary sequence in order to make the protein. So both conditions exist in different viruses, right? So that's why they rely on sometime on reverse transcriptase, sometime on RNA-dependent RNA polymerase. So a little bit complication there, right? But I hope that you understand the major, like main idea that why you need sometime you have to make RNA to RNA and sometime RNA to DNA, and then from DNA to again you have to make RNA. So during when they are making again RNA, that time they are synthesizing the correct thing from where protein will be synthesized. Is it clear?
Yes, sir.
Yeah.
Yes, sir.
Like other student, like Soham.
Yes, sir.
No, I'm saying that this RNA to RNA or RNA to DNA, you understand, right? Because
Yes, sir.
I'm not going into details, but this idea should be there. Okay, so uh, let's see. Now we should go to the next slide. Here, virus life cycle, I will very briefly discuss about the life cycle. But as I told you, we are not going to ask you the details of how virus replicate or virus life cycle, but you should know those things. So here, this is one virus particle, and if you see the outside layer, this is capsid, right? So this is capsid or protein layer here. This is capsid. And now some proteins, they are present like this and like this one also. And this is actually spike protein, right? And inside the virus particle, this thing is actually DNA or RNA, whatever the virus is, either DNA or RNA. That means genetic material. And here spike or spike protein that helps to attach with the host cell. So viruses, they are actually specific to the
All viruses will have
Mhm.
spike proteins, or all viruses will have spike proteins, or only some viruses have spike proteins?
Yeah, so some I would say like viruses, most of the viruses, they if they have the envelope, they don't rely on spike protein. Okay, so but other viruses like capsid containing viruses, they have some protein like spike-like structure, and that helps to anchor or help to interact with the receptor of host cell. So I would say spike itself a component of capsid. Okay, the spike itself a component of capsid layer. So spike is also a protein, and but in order to answer your question, no, all viruses they don't have spike. Some viruses they have spike. Clear?
Yes, sir. Then how does the other viruses are here the receptor is actually connecting to the spike protein? Then how do the other viruses which do not contain the spike
Yeah,
protein actually?
yeah. See, the term spike protein means it's present as spike, that's kind of spike. But if you ignore that, as I told you, that some viruses with envelope, they can fused with host cell membrane, and the virus particle they go inside. So that time spike protein is not required. That is one example. Another example is some viruses, they just sit on the host cell, and they have some injection machinery. They inject their host genome, their viral genome inside the host cell. Not the capsid or like those things are not going inside the cell, because what present inside the virus, the major thing is nothing but the protein layer and the genomic material. Because if the genomic material or the genetic material that is going inside the cell, it will make more and more virus particle, right? So that is another strategy how virus infect host cell. So as a result of that, all viruses they don't rely on spike protein. Clear?
Yes, sir.
Excuse
Yeah,
me, sir.
yeah.
Sir, the envelope is uh, the virus has envelope from the host organism only, no? So how is
Yeah,
it present from first?
yeah, from host only. Yeah, because virus they don't have information to make envelope. Hmm?
So first it comes contact with the plasma membrane of the host, then it kind of uh, wraps it around itself and then enters the host cell.
Yeah, exactly. Because lipid they can integrate. So a virus particle, they if they have the envelope layer, they finally integrate with the host cell membrane, and then the protein part or the viral particle without envelope. Envelope will be integrated with the host membrane, and the virus particle go inside the cell. That is another way of infecting host cell. So there are many different machinery. As I told, even fuzz virus, they inject their genomic material only. So here you can see that when this virus that get attached with the receptor, and then some entry mechanism, here again there are different types of entry mechanism. In many cases what happened, host cell itself phagocytes, like that is called endocytosis. That means the virus particle and the receptor together, it will come inside the cell. It will come inside the cell together, the receptor as well as the virus particle. So I am not going into the details, but that's why we say that virus hijack our receptor, but host cell will not understand this is virus. So whenever this receptor will be bound with some particle or virus particle, then it will get endocytosis. Endocytosis means it will go inside the cell, and then virus is inside the cell. Then what will happen? Then uncoating or disassembly, and then translation, replication. It depends on what kind of viruses are those, DNA virus or RNA virus. Like as I told you, RNA virus can be of different type. So finally, if you see that from the viral genome, you can get mRNA, and then from mRNA you will get protein, and from those protein you will get the capsid layer, like proteins which make the capsid layer. And similarly, like when those proteins will be ready, in the in the same time, from this viral genome, replication is also happening, and you will have more and more viral genome, and finally they will package together, and finally those viral particle will come out from the host cell. Here I would like to mention, at your level, just we will discuss something else. So don't concentrate here, like particularly with those RNA virus, DNA virus, negative and positive sense RNA virus. As I told you that two types of different types of RNA viruses. So just try to understand that what happened here in case of viruses. When virus infects host cell, they go inside the host cell, and the genetic material of viruses that contain the minimal information that will make some important thing. For example, the protein, capsid protein, which are required, right, in order to make more and more virus particle. Also, like some enzyme which will make the which will synthesize its own genome. For example, if some virus they have RNA as their genetic material, and then from that RNA they will make more and more RNA. Then they will rely on RNA dependent RNA polymerase. But we don't have in our cell, in human cell, we don't have RNA dependent RNA polymerase. That means that information is present inside the virus genome itself. So then what will happen? That virus genome, from that genome, the translation machinery of our cell will make some protein, and some of them will be this RNA dependent RNA polymerase that will help to make more and more RNA, right? See, two things are happening. One way, you are getting more and more genomic material, whatever they have DNA or RNA, ignore that part. On the other hand, you are making more and more protein, like that include capsid protein as well as, I know, capsid protein is the main thing definitely. And finally, inside the cell, you have huge number of viral genomic material as well as those protein, and they will assemble together, and at a point of time they will come out from the cell. So virus are coming out from the cell, and just from one or two viruses you will get huge number of viruses at certain point of time. This is how virus infect us, and then after assembly of their own particle they actually leave that cell and then again infect some new cell, right? This is how viruses they infect host cell. So here, virus growth. I will actually, honestly, I would like to touch all, like, like basic aspect of this thing, as well as some of the thing, like, even how do we test like viruses that count, like counting viruses, that like many people actually like this time, because of this pandemic, suffer actually, some of our family members, like many of them. So sometime when you test, like for example, RT-PCR, they provide you some value, that is how much, whether the viral load is too high or viral load is too low. And as well as, apart from that, in lab, we count virus in a different way. Okay, so those things also I would like to, I would like to explain a little bit, so that you will get like, some kind of overall idea. So let's see. So viral growth or virus growth. So in this case, in case of bacteria, I told you
So
that
the
bacteria from
slide
one.
is not appearing. So for me the slide is not now it is appearing.
Okay, so you can see the virus growth, right?
Yes, sir.
Okay. So as I mentioned previously that, like, in case of bacteria, what happened, if you have the suitable environment, you have food. So from one bacteria, in the next round, you will get two bacteria. In case of E. coli, this is just 20 minutes. In other bacteria, it can be different time. Again, from two bacteria, like here. So it is going like this, right? Another 20 minutes here. So as a result of that, you normally get this kind of curve. So initially it is going a little bit slow, then it is log phase. That means, bacteria, they are dividing properly. Then in the container where you are growing bacteria, like either food is low or number is too high. So then the growth is a little bit reduced. They are not growing anymore, something like this. Okay. So this is bacterial growth kinetics, how bacteria grow. But in case of virus, it's completely different. I believe that you can understand by your own if you listen properly my, I know, explanation in the last slide. What happened here, in the lower panel, this is the viral growth. So in y-axis, this is number of infectious particle, that means number of viral particle, and in x-axis, we are showing time. Okay, it can be hours, it can be day. Now, if this is the starting point of the infection, and if it's in the lab, then I would say this is the starting point of the viral culture, right? You are trying to grow virus. So first of all, tell me how I can grow viruses in the lab. So I told you that several times in Michelson style experiment, in many experiment, that you can make some kind of liquid media and you can put some bacteria, and in suitable condition, like, proper temperature, bacteria can grow properly. So how we can study viral growth inside the lab, in the lab? What do you think?
So we need some ribosomes for them.
Yeah, then, that means, yeah, ribosome, in a separately, it is not possible. If they have ribosome, but still they have, many things are missing actually. So
We need
ribosome
the
is one of
host
the
cell
important
as
things. Very good.
we
Yeah.
need the
So
host
that
cell.
means, yeah, exactly. So that means here media means not like media like bacterial growth. Here, in order to grow viruses, first you have to have some kind of specific host cell. That means some virus, they infect a particular type of host cell. Like, so you need host cell in the lab first. Then you have to infect host cell with the virus particle. And then those virus will attack host cell. They will actually make more virus particle that you have to measure, right? So this is the difference. You need host cell in order to grow virus in the lab. As I told you before, that without host cell, outside the host cell, virus cannot be, multiplied. So that's why we cannot see any growth of viruses outside the cell. So here, that's why you can see that when you start this infection, that means you have some host cell maybe, and or it is, if it is in the body, that means someone just get infection. Then sometime it can be few hours from some viruses, it can be few days. That is, you will not get any increase in number of virus particle or infectious particle. This is called eclipse period. And then suddenly you will see a burst of synthesis or burst of
So
yield.
the eclipse period is also the same as the incubation period that they say.
Yeah, exactly. Like, it's right. So in normal, like, mostly when you see in news channel or yeah, incubation time, you are absolutely right. I will come to that also, right? So then burst of yield, like that means like huge amount of virus particle they are coming at the same time. You can see the number going inside, like, growing very high like this. Then the reason is, if I ask you what is the reason, I believe most of you can actually like, I believe that most of you have the answer that why it's happened like this. Who will try? Because see, like
Hello, sir.
bacteria, yeah,
Sir, because the spike, sorry, the capsid and the genome material starts to fuse with each other, and the number
yeah,
of virus particles starts to rise. Right.
exactly. So in case of bacteria, what happened? Even in case of our cell, what happened? Whenever the suitable environment there, our genomic material get double, immediately the cell divides. Right? It is true for bacteria as well as in human cell. But in case of virus, what happened? I explained in the life cycle that it infects host cell, it go inside the host cell, then it will make huge number of viral genome inside the cell. They are using host cell as just factory to make their own gear and own parts. And then they are also making lot of proteins like capsid proteins. Whenever like they have like utilize most of the resources inside the host cell, then they want to survive, they want to infect more and more cells, so they get assembled and they come out from the cell. Someone asked about incubation time. Yes, this is true. So the thing is, in some viruses it takes like 3 days, 4 days, 5 days. So for example, in case of SARS-CoV-2, if you see the like median, I would say the average time, incubation time is around 5 days. Right? In some cases, it can be 2 to 3 days, sometimes more than 7 days, but the average time is 5 days. That means someone infected with some viruses, right? Whether this is SARS-CoV-2 or any other viruses, immediately 1 hour later, 1 day later, if you try to see whether the infection is there or not by some like some kind of test, you might not get anything because the particle number is not yet there, no virus particle anymore. Just genome is inside. They are synthesizing all those things. Whenever they will actually make all those particles and they will come out from the cell, they will infect other cell, then only you will start getting the number, right? This is like the minimum time you need to after that only if someone like carry out the testing, then they will get some result. Okay, so now this is how a viral growth curve look like. Then what will happen? Then I would say again, see when this like viruses are coming out from the cell, then you have
So,
burst of
can you
it.
give an approximation of the like from one host cell, how many viruses can like on an
Uh,
average can
Honestly,
you
I
give?
would say like I don't have this like the right answer, but I would say definitely it more than thousand, not thousand, right? But it's hard to say why I'm telling you because it depends on what kind of virus you are talking about, whether they are infecting lung cell or some other cell. Because like in our body, some cells are really big compared to other cells. Some viruses they are infecting epithelial cells, some viruses lung cell. So I'm sure that it will be drastically different one cell to another cell, even one virus to another virus. But definitely the number is not like from 1 cell to 5 virus or 10 virus, not like that. It should be like 100,000 something like that in number. Okay.
Okay, sir, got it.
So, say for example, lung cell like our alveolus, those are relatively much bigger cell. They have more space also, right? So it depends on many things. And now here, so I should like little bit elaborate this. So here after infection, if you consider just here one cell, one host cell, and here one virus particle enter inside this host cell. Just for explanation, I'm saying one virus particle went inside this host cell. Then it is taking time, and from this one host cell, as you ask that how many viruses, I would say, okay, few thousand viruses coming out. Then those few thousand viruses are here, and now they are actually infecting many cells in our body. Then again they are taking time, right? And then again you will see another round of like burst of yield. So it this way it can grow. And finally, you will not see this kind of thing. Either the viral burden will be too much to handle by the host cell or the host organism, or by that time, like our immune system that can detect the virus and it can dampen, like it can defeat the virus also. That is also true. But virus growth is like this. And now, how to measure virus titer? That means how to measure the number of viruses. This is in the laboratory. What like most of the virologists they do in the lab. This is very classical technique. RT-PCR thing I will come later at the very end of this lecture. But here this is classical experiment and done in 1952, and he got Nobel Prize in 1975. So this assay, this is called Plaque assay, it gives the measure of infectious virus unit per milliliter of sample. And it depends upon the ability of viruses to infect and kill cells. Okay, so that means whatever we are counting the number of viruses, we have to assume that all viruses are infectious particle and they can kill the host cell. It doesn't account for the non-infectious or defective virus particle, right? It is only considering the virus which are active and that can kill the host cell. So let me explain. I think within 2 or 3 slides, I will explain the old is go. I would say this is kind of old strategy, but still virology lab, like we were working with virus in different lab throughout the world, they still use this technique. This is called Plaque assay, okay, to get the number of viruses. So let's see. So Plaque assay means actually, normally the way I explain, most of the information I already given you, even today's lecture, that how you will grow virus. First thing is you have to have some host cell in culture. So this is called, so here what you can see here, this is called Petri plate. Okay, P-E-T-R-I, Petri plate. So some kind of, like glass plate. Here you have host cell. So this is, so for example, some human cell, right? This is host cell. So cells are seeded into cultured dishes so that they can form monolayer. These cells mean not viruses. Virus, I will not say virus cells. Cells means host cells. So in the lab, we have different types of cell line. That means even, say, for example, in my lab, I have some cell line that is, alveolar cell line, right? Although I'm not working with viruses at all. So, but for different purpose, I have some lung cell line, alveolar cell line. So that means those cells I stored at, in liquid nitrogen. So, and whenever I want to do some experiment, what I need to do? I have to take some of the alicate of that cell which is present in liquid nitrogen at low temperature. You have like correct procedure. You have to thaw that cell. That means from very low temperature to room temperature. And then finally, I have to put that on that petri dish, whatever I just show you. And in the petri dish, you should have some kind of liquid media. That means again, some food, some other suitable environment for cell. But this cell line means this cell can grow actually. In proper condition, this cell can grow in number also. Okay, that is possible. And this is how we grow mammalian cell in lab. But what I want to discuss here a little different. Our goal is to understand how we can get the viral load or the number of virus particle present in some sample, right? So in order to do that, first we culture host cell in monolayer. That is very important. Monolayer means you are diluting your sample in such a way that the host cells are present in this Petri plate in single layer. How you will confirm that? After putting those cells in this Petri plate, you can observe that in microscope. And you can actually, if you are experienced, then you can be assured that yes, your cells are in monolayer. That means just single layer there. Okay. And then the next step is the tenfold serial dilution of virus stock solution. Say, for example, you have taken some, I would say, some water sample, right? I'm just, all the time, I'm not going to say that, talking about SARS-CoV or human viruses. Say, for example, some water sample, and that is, that is containing some viruses, and that virus can also infect some host cell, some mammalian cell. So then what will happen? This is the virus stock, some water sample you have taken where some viral contamination happened. Then you have to make some serial dilution. Say you are taking from this solution, you are taking just 0.1 ml, that means 100 microliter here, and you are adding 0.9 ml of dilution buffer or water, you can say here. And then again 0.5, 0.1 ml here, then again 0.9 ml water, 0.1 ml here. So this is called serial dilution. All of you are aware about what is serial dilution. What's the purpose? You will know why we are doing this. So after this serial dilution, that means higher dilutions are used to make sure one cell get infected with one virus particle, right? That means when it is like the dilution is really high, that means if you take like few microliter sample from here, then it assure you that just one virus particle there. But again, we have to carry out some experiment and then we will see if it is correct or not. Let's see. So if this is the case, when you have diluted this sample too much, then you are taking, for example, here 10 microliter sample from this tube. And from this 10 microliter, if you have just one virus particle, then what will happen? That virus particle will infect one cell here in this Petri plate, right? And this one cell, what happened? That virus particle will go inside the cell, and virus particle will, like make more and more virus particle inside the cell. But here, some important thing: after the infection, that means after adding the virus, the cell's monolayer are overlaid with agarose media, some semi-solid media or mixture that forms a semi-solid layer. This inhibits the newly formed virus to come out into the media and infect other cells at a distance. Very, like, I'm trying to summarize again so that everyone get the point. In this Petri plate here, you have a single layer of host cell. Somehow you diluted the virus particle here, the virus stock solution, through serial dilution. And if you are giving virus particle from the highest dilution tube, that means you have the lowest concentration of viruses, just little amount of this solution here. And if you consider just one virus there, so it will infect this cell, right? And I told you after some time, after few days, it depends on the virus. So this virus particle will destroy this cell, and then destroy means they will grow inside the cell and they will come out from the cell. But the problem is, if those viral particles are coming out from the cell and they are infecting another cell present here, then you will be you cannot calculate the number of virus particles. So that's why what we do after this infection, that means after giving this virus particle here, we put some semi-solid layer of agarose so that although virus particle will be generated here in this cell, and after they are emerging out from this cell, they cannot travel to here. They cannot travel long distance. Only they can infect nearby cell, maybe just adjacent cell, like whatever cell present here, just one cell, two cells, just those adjacent cell. That's all. They cannot infect any other cell. So as a result of that, if that is the case, then you can say that in this solution, whatever you have taken, you had just one viral particle. I will show you one real data, like real experimental data, and then I will ask you whether you understood or not. So here, plaque formation in a monolayer cell. For example, if the virus particle is here, right, and this is the host cell. Host cells are much bigger than virus particle. Whatever the scale I am showing here, this is not right. Host cells are even much bigger than the virus particle. And now this virus particle, when synthesize its material inside the host cell, they can, some of those come out from the host cell and they will infect the surrounding cell, but they cannot travel long distance to infect other cell which is present in the plate, because I told you we actually on top of that we put some semi-solid media just to prevent this thing. So as a result of that, over the time this virus particle will kill those cells, and as a result of that, those cells will not be there. Those cells will be lysed. That
So
means those.
I have a doubt.
Yeah, sure.
So why do the cells get killed? Like they are only producing the viruses in a large number, and that they do for all organelles within the cell itself. So what exactly kills the cell?
Yeah, good question. I didn't tell that actually, because finally virus will, come out from the cell, right? How they will come out? That is called the lytic cycle. Lytic is coming from the word lysis. That means rupture of the cell. So whenever the virus will come out from the cell, they will completely lyse the cell. The cell will get ruptured. The cell will not be there anymore. It will rupture. So that's why the cell is, clear, Tanmay?
Yes, sir. Yes, sir.
Yeah, so cell will get ruptured. So now we have some staining procedure. If we, use the staining procedure, then we will see all the live cells. They will take some dark blue stain. I'm not going into that details of staining. You don't need to know. Like just you have to remember multiple steps. It's not required. This is called crystal violet staining. But anyway, like all live cell will take blue stain, but here in the center you are not seeing any blue thing. You will not able to see any virus particle. As I told you, this experiment we are conducting in light microscope, and I told you in order to see virus particle you need to see through electron microscope, right? So you will not see any virus particle, rather you will see some white or translucent spot here that is not blue in color. That is called plaque. Okay, so that means if you have one plaque here, that means in that solution you have just one viral particle. That's why you get one plaque. So let me go through one more slide, then I believe you will get it. So this is what you can see here. This is a small portion of that Petri plate, but you can see the similar thing in a different region of the Petri plate also. See here, is actual experiment. It's real experiment. Actually, one of our colleague, actually a virologist in our School of Bioscience, right? So I'm taking this from his lab itself. So here you can see the different dilution. So 10 to the power 2, 10 to the power 3, 10 to the power 5, 10 to the power 4. So that way, different dilution. So here, instead of Petri plate, we are using a different type of, you can see that here, 12 oil plate. 12 oil plate means in this like one big rectangular plate you have total 12 oil kind of thing. You can consider that this one is one Petri plate, this one is another Petri plate. You can consider that, right? Now in all these oil or all these depression, whatever you can see, you have the monolayer of same host cell in same concentration, everything same, the host cell there. Now what happened? Here in this first two oil, right? In this two oil, you have added very much concentrated stock solution. Then it is little bit diluted. Then little bit diluted here. 10 to the power 4, 10 to the power 5, 10 to the power 6, 10 to the power 7. Now see, after incubating, say for example, if it is 5 days, then the staining procedure conducted, then what you are observing? I'm just explaining. I believe you will get it. So for example, here where you added concentrated viral stock solution. So here you can see nothing blue. That means viral concentration is too high that virus particle actually infect all the host cell present in this oil. And as a result of that, nothing blue you can see after staining. Here also similar scenario. Now see in this dilution, what happened? In this case, uh, here you can see some blue and white mixed together, but you cannot count any number of plaque. So now you have to go down, right? You are actually taking more and more diluted samples. Say, for example, in this sample, in 10 to the power 6 dilution, like you are diluting, taking from this tube to next tube, that way. You can say 10 to the power minus 6 actually also. So now, if you see here, here you can see 1, 2, 3, 3 spot here. Whatever I explained in the previous slide, that means one virus particle was there and it makes a plaque, another virus particle here. So that virus particle infect one cell, from that cell surrounding cell also get infected, and finally after staining we see like here no host cells are there. Blue things means host cells are live and there, right? So as a result of that, three plaque formation in this plate or in this oil. So that way you can actually calculate the number of virus particle present in the solution from where we have taken to infect these two oil, right? So this is the idea how it works. So for example, here, this is from some textbook or some paper, so that you will get a better idea actually. That was the real experiment. So you can see when it is less diluted, you have many plaque formation here. You cannot count. When you have very less dilution, like very high dilution, you have only two plaque. And here you have like reasonably good number of plaques what you can count, but it is not too low. 17 plaque here in this concentration. They are taking 0.1 ml of sample from this tube, and they are getting this number, and you know that how much you have been diluted this sample. So that way you can calculate what will be the number of virus present in this stock per ml or per microliter that way. So this is the idea how people used to calculate the number of virus particle inside a host cell, not host cell, like in the lab. And this is very classical thing, and still people are doing, because the number is absolute. Here, I would say the mistake is much more. I would say the number is very precise actually. So the counting method is more reliable. So when you are doing some critical experiment to understand something, people use this Plaque assay. Okay. So now you can take break for 3-4 minutes, and then I will come again. If you have any question, quick.